Hi, in this video we will discuss about the Toll-like receptor basically. So the Toll-like receptor are present in the immune cell, which that is the first line of defense. Basically the macrophage or dendritic cell contain a Toll-like receptor and also node-like receptor. So first of all, in this video we will discuss about just a Toll-like receptor, and next video we will discuss about the node like receptor which that is present in the cytoplasm. So the toll like receptor are present in the plasma membrane, and which that is recognize the different type of pathogen and their protein or antigen or epitope you can say. So this is the basically is the major properties of the macrophage, which recognize the bacterial or viral epitope, and after the recognition, so the endocytosis will be occur, and this endocytosis can be bacterial DNA, RNA, or single stranded, double stranded, as well as other things. So we will also understand about how they work and what is the number of the toll-like receptor we will discuss in this video. So let's start to understand the toll-like receptor. So let's begin to understand. Here is the basically the pathogen first of all we will discuss. So this toll-like receptor is TLR is the short form, and the first line of defense. It means the macrophage and dendritic cell contain a toll-like receptor. So in this way here you can see the pathogen. This pathogen can infect the tissue, and this is infected cell produce the inflammatory cytokines. For example, interleukin 1, tumor necrotic factor alpha, and tumor necrotic factor gamma. These tumor necrotic factor alpha and interleukin 1 and tumor necrotic factor alpha, which stimulate the monocyte, are present in the red blood in the circulatory system. So after the monocyte will be proliferate to form a macrophage. This macrophage is the special character due to the monocyte contain a nucleus and nucleus contain gene. This gene is activation from the toll-like receptor gene produce the toll-like receptor on the plasma membrane of the macrophage. So after this plasma membrane on the macrophage will recognize by the antigen with toll-like receptor. After the recognition will endocytosis occur, and the macrophage will endocytose the antigen, for example the pathogen, bacteria or virus. So after this is the endosome formation, and this endosome formation, the hydrolytic enzyme and the lysozyme enzyme are present, which that is convert the pieces of the antigen, which convert and donate the by MHC molecule to donate and recognize with the T cell, naive T cell to activate by the T cell receptor and CD4. This is the CD4 helper T cell, and on the other hand also produce activate the cytotoxic T cell for the killing of the infected tissue. So in this way you can see the CD8 basically produce the proliferation and the recognition of the epitope via a CD4 molecule, and this T cell also produce cytokines to more proliferation and recruitment of the monocyte also from the circulatory system, and in this way the monocyte will be more proliferate to produce the macrophage, and after this macrophage production that will be increase the endocytosis of the pathogen. So this is the first line of defense, but it is also related to the adaptive immunity due to the production and stimulation of the B cell to produce antibodies. So let's begin to understand the toll-like receptor. So here is the peptidoglycan or bacterial, basically is the different we will discuss. So here is the dimeric form of the toll-like receptor 1 and 2. So the toll-like receptor 1 and 2 are present in the dimer form. Di mean 2 and mer mean the mer's part, and in this way the two toll-like receptor 1 and 2 are present in the same situation, same collectively couple type. So here you can see the dimer, another is the toll-like receptor 2 and toll-like receptor 6. This toll-like receptor 2 and toll-like receptor 6, which basically is also in the form of dimer, and basically 10 in human, but the 11th are present in the mice. We will not understand about that. So here is the 10 in human, but the 11th is the not functional in the human body. So here you can see the toll-like receptor 5, which that is the monomeric, and this is the monomeric, and here is the toll-like receptor 11, which that is also monomeric. So the toll-like receptor 10 is also present, which that is recently discovered, and check the GP41 protein from the HIV virus. So here you can see the peptidoglycan and bacterial gram-positive negative bacteria and lipoprotein and zymosin in fungi, which that is recognized by the toll-like receptor 1 and 2 as well as the 2 and 6. So the toll-like receptor 4 is dimer, but both are same. So it is the homodimer, so not heterodimer. And in this way, the lipopolysaccharide of gram-positive bacteria will detect and toll-like receptor through a toll-like receptor 4, while the flagella is detection through a toll-like receptor 5, and the toll-like receptor 11, which that is the uropathogenic profilin, grass bacteria, and wheat in mice, which that is detect from the bacteria. So here is the intracellular part, and here is the plasma membrane. So the one is the 3 and 7 and 8 and 9, which that is basically where is the that. So that is present in the cytoplasm. Basically the 3 and 7 and 8 and 9, which that is present in the cytoplasm. So here you can see that is a fungi, bacteria, the double-stranded RNA, CpG RNA, DNA, and single-stranded RNA. So the unmethylated DNA also will detect of the viral DNA, and after the endocytose by a endosome formation. So this endosome formation will move inside by a DNA and RNA containing. So the toll-like receptor 3 and 7 and 8 and toll-like receptor 9 is collectively detect the. So the toll-like receptor 7 and 8 detect the single-stranded RNA, while the 9 is CpG RNA, DNA, and the toll-like receptor 3 is used for the detection of double-stranded RNA of the virus. So after this, all this activity will be recognized and initiate a cascade of events. And this initiate cascade of event is very difficult to explain, but here I am giving the overview. So that is very complex situation. We will not understand that. So here is the activate certain transcriptional factor. This activate certain transcriptional factor can be three types. AP1, this is the activated protein 1, and the IRF protein interferon regulatory factor, and the necrotic factor kappa B. This is a three type which that is through a activated certain transcription factor. This is the three type of transcription factor which that will be bind with the DNA in the nucleus. So here you can see the membrane of the nucleus. So the nucleus is membrane, nuclear membrane. So in this way you can see that is the activated protein 1, which that is bind with the DNA with a specific gene where the transcription will be occur, and the produce messenger RNA, and each produce the messenger RNA on the basis of the gene activation IRF and AP1 and necrotic factor kappa B, and each produce the messenger RNA, and that messenger RNA will go outside from the nuclear pore. So you can see the transporting protein or something. So in this way, here is when move outside so the messenger RNA, for example, so begin into the endoplasmic reticulum, rough endoplasmic reticulum, where contain a ribosome, and this ribosome is basically the AP1, which that is produce the different differentiation and proliferation of uh, apoptosis. So this is important and used for the apoptosis and the differentiation and proliferation. So on the other hand, the messenger RNA, which that is produce IRF, which that is IRF transcriptional messenger RNA will produce the interferon INF, and on the other hand, the necrotic factor kappa B messenger RNA containing produce the pro-inflammatory cytokines, for example, tumor necrotic factor alpha. So these all things produce the tumor necrotic factor alpha pro-inflammatory will be activated to become a inflammatory. So the enhanced immune response via a TNF alpha. So on the other hand, the pro interleukin 1b and the pro interleukin 18, which that is inactive, and in this way the inactive pro interleukin 1b and pro interleukin 18, and the proteolytic cleavage for pro interleukin 1b and pro interleukin 18 to become a maturation and release of cytokines. So in this way the pro become a mature interleukin 1b and interleukin 18 through a caspase 1 protein. This is the proteolytic cleavage protein caspase 1. This caspase 1 is used for it for the proteolytic cleavage. So after the cleavage formation to produce the mature cytokines interleukin 1b and interleukin 18, and this will be also the interferon will be also move out from the plasma membrane, as well as the another is the interleukin 1b and interleukin 18 also. And after this bind with a single cells receptor signals to other cells about pathogen and to increase the defense. So the single receptor contain a single interferon. So in this way, the signals to other cell about pathogen and to increase defense. And in this way, the protection, also the recruitment through a cytokines produce itself this tissue to recruitment of the immune cells. For example, monocyte, T cells, B cell for second line of defense, or adaptive immunity through a innate immunity. So the macrophage also produce and more recruitment. And in this way, the cytotoxic T cell will kill the damaged tissue, and neutrophil also produce for the quick response during the pathogenesis formation. So this all cell, monocyte, T cells, B cell, macrophage is important for the immune response and neutrophils, you can see. And I hope you make sense about the toll-like receptor. This is toll-like receptor. Why is known as? Because it is look like a toll. So that is important things you know about that. So anything is related to the pathogen which that is recognized through toll-like receptor, and after the recognition to endocytosis of the pathogen, as well as also produce the cytokines for recruitment of the other immune cells, and in this way the macrophage plays dual role, endocytosis as well as the recruitment. So this is the important thing. So thanks for watching. Bye.